Welcome back to the deep dive. Today we're tackling something that's, well, honestly, it's one of the most frustrating paradoxes in modern medicine. Think about it, organ transplantation. It's just incredible, right? The idea that, one person's kidney or liver, even their heart, can essentially restart another person's life. It's a massive achievement, scientifically speaking. But there's always a but, isn't there? Lurking right behind that miracle is this huge, ongoing, and potentially really dangerous problem.
That's exactly right. The surgery itself, it's life-saving, no question. But the price patients pay afterwards,
Yeah.
it's steep. We're talking about a lifelong dependency on broad-spectrum immunosuppressive drugs.
And these aren't just like taking a daily vitamin. These are powerful chemicals designed to keep the entire immune system basically dialed down permanently.
Yeah, and powerful often means toxic. These traditional drugs, your calcineurin inhibitors, antiproliferatives, they create serious systemic issues. It really impacts quality of life, sometimes even lifespan.
So when we say toxic, what are we actually talking about? What are the big risks?
Well, the risks are significant. You have a much higher chance of getting serious, even life-threatening infections because your immune defenses are lowered. There's also an increased risk of developing certain cancers. The immune system normally helps keep those in check.
Okay.
And then there's long-term damage, like cardiovascular problems.
Yeah.
And here's the real kicker. Despite taking this cocktail of powerful, toxic drugs for life, they often still don't completely prevent the slow, creeping damage of chronic rejection.
Wow. So you endure the side effects, but the original problem might still happen anyway.
Precisely. It puts doctors in this incredibly difficult position, suppress the immune system enough to protect the new organ, but not so much that the side effects or an opportunistic infection become overwhelming. It's a constant tightrope walk.
You mentioned this has been known for a while.
Oh, yeah, since the middle of the 20th century, really. After Peter Medawar's work in the 1950s showing you could actually induce immune tolerance in animals, the holy grail for transplant medicine became clear. Achieving donor-specific tolerance. Basically, getting the body to accept the transplanted organ completely without needing those lifelong harsh drugs.
That must have sounded like, well, science fiction for a long time. How do you possibly convince the body's security system that this foreign thing is actually okay?
For decades, it really did seem impossible. But then, researchers started finding clues. They discovered the immune system has its own internal police force, its own peacekeepers, a completely new way to think about control.
Ah, okay. And that leads us to today's topic.
Exactly. The discovery and the deep dive into understanding regulatory T cells, or Tregs, this fundamentally shifted everything. Instead of just hitting the immune system with a chemical hammer,
Right.
we started thinking maybe we can harness the body's own system of control, use its natural mechanisms to achieve that tolerance we've been searching for.
And that's what we're diving into today. We're going deep into the science that offers this, well, this new handle, potentially, on curing transplant rejection. We'll look at the crucial discoveries that first identified these cells, unpack the clinical trials happening right now, and explore the really cutting-edge stuff of cellular engineering, CRR technology, synthetic growth factors, all the things that might finally make drug-free transplant survival a reality for people. #2, the groundbreaking discoveries.
So to really get why Tregs are such a big deal therapeutically, we have to go back to how they were discovered. Because honestly, it changed the whole way we thought about how immunity works. For a long time, the focus was all on the on switch, how does the immune system attack?
Like it's just about activating the attack dogs, right?
Pretty much. But Tregs showed us there's a very sophisticated off switch, too. These Tregs, they're like the immune system's internal security detail, specialized in enforcing peace and preventing the system from attacking itself, or in this case, the transplant. The real breakthrough came in 1995 from Shimon Sakaguchi's lab in Japan.
Okay, Sakaguchi 95. Tell us about that experiment. What did he actually do?
He was working with mice, taking apart their immune systems, essentially. He focused on T cells, the main orchestrators. He found that if you took out a specific group of T cells, the ones expressing a marker called CD4 and then transferred the remaining T cells into mice that lacked an immune system,
Okay.
those recipient mice developed absolutely devastating autoimmune diseases, multiple organs attacked, a total failure of self-tolerance. The immune system just went haywire against its own body.
So removing that one specific cell type unleashed chaos.
Complete chaos.
Yeah.
Sakaguchi called them sick mice. Without that CD25 positive group, the other T cells were just uncontrollably aggressive. And this is the crucial part, if you took those CD25 positive cells he'd isolated and added them back to the mix before transferring them.
Let me guess, the mice stayed healthy.
Completely healthy. It proved these CD25 positive cells weren't just hanging around. They were actively, forcefully suppressing that destructive immune response.
Wow. So it wasn't just the absence of attack signals, it was the presence of active calming signals.
That's the core idea of active immune regulation. It showed tolerance is an active process maintained by specialized cells. It shifted the whole field. Instead of just thinking about how to chemically dampen the immune system, people started thinking about how to biologically boost this natural calming mechanism.
That is a fundamental shift, but knowing a surface marker, CD25, that's one thing. Figuring out what makes these cells tick genetically, that's another step, right?
Absolutely. The genetic key came a few years later. Two research groups, led by Mary Brunkow and Fred Ramsdell, were working independently on these really severe autoimmune syndromes. One was in mice, a fatal condition called scurfy. The other was in humans, a devastating disease called IPEX syndrome.
Scurfy mice and IPEX humans, what was the connection?
They both boiled down to mutations in one single gene, FoxP3. Brunkow and Ramsdell discovered that FoxP3 acts like a master switch, a transcription factor that's essential for developing these regulatory T cells. If that FoxP3 gene is broken,
Whether you're a mouse or a human.
you simply can't make functional Tregs. The body lacks its peacekeepers, and the immune system essentially self-destructs.
So FoxP3 isn't just involved, it's the critical gene, the defining characteristic of a Treg.
Exactly. It's the master regulator. No functional FoxP3, no functional Tregs, no immune tolerance. So putting it all together, Sakaguchi showed these cells existed and actively suppressed, and Brunkow and Ramsdell identified the genetic master switch, FoxP3, that defines them. It laid the entire foundation for understanding how the body maintains peace.
And it's worth pointing out, this wasn't just some incremental finding. The combined importance of this work, identifying Tregs, defining FoxP3, proving active tolerance, was recognized at the highest level. Sakaguchi, Brunkow, and Ramsdell shared the 2025 Nobel Prize for this.
Right. It underscores just how fundamental this discovery was. It didn't just open doors in transplantation, it potentially revolutionized how we think about treating all kinds of autoimmune diseases, too.
Okay, so we know FoxP3 is the command center inside the cell. But what does a Treg actually look like phenotypically? How do scientists pick them out of the crowd of other immune cells, and what's their basic job description?
Right, so phenotypically, Tregs are a distinct subgroup within the CD4 positive T lymphocytes, the helper T cells. We identify them by a specific signature of markers. The key ones are high stable expression of that FoxP3 protein inside the nucleus.
The master switch.
Exactly. And high expression of CD25 on the cell surface.
CD25 Sakaguchi's marker. Now we understand its significance better.
We do. CD25 is the high affinity receptor for a growth factor called interleukin-2 or IL-2. Tregs have lots of it, which means they're really good at grabbing onto any available IL-2. They effectively soak it up.
Okay, so high FoxP3, high CD25, anything else?
Yeah, there's a third important one. Low expression of CD127, that's the receptor for another cytokine, IL-7.
And why is low CD127 important?
Well, IL-7 is usually a key signal for survival and expansion for conventional, potentially aggressive T cells. By having low levels of the IL-7 receptor, Tregs are kind of less tuned in to those general pro-survival signals. It helps distinguish them from the effector cells that might cause rejection. So that combination, high FoxP3, high CD25, low CD127, that's the classic Treg signature we look for.
And their function, as the IPEX syndrome tragically shows, is absolutely critical.
Indispensable is the word. Their core job is maintaining immune balance or homeostasis, and particularly self-tolerance. Without functional Tregs, as we see in FoxP3 deficiency, the immune system runs rampant. So they are fundamentally essential for preventing the immune system from attacking self, and by extension, for preventing it from attacking a transplanted organ which immunologically the body sees as non-self. Boosting them just seems like the most logical way to encourage acceptance. #2. The redundant suppression mechanisms, how they silence attackers.
One thing that really stood out to me from the research is that Tregs aren't just a one-trick pony. They use multiple overlapping strategies to shut down an immune attack. It's like they have this whole toolkit dedicated to creating a suppressive environment locally.
That redundancy is really key to their effectiveness. If one mechanism isn't enough in a particular situation, or if the attacking cells find a way around it, another mechanism can kick in. It ensures they can handle different types of threats. We can broadly group their methods into two categories: contact-independent, using signaling molecules, and contact-dependent, which involves direct cell-to-cell interaction.
Let's start with the signaling molecules, the contact-independent stuff. Cytokines, right?
Right. Tregs are factories for potent anti-inflammatory cytokines. The big three are interleukin-10, IL-10, transforming growth factor beta, TGF-β, and another one called interleukin-35, IL-35.
Okay, what do they do?
IL-10 is sort of a classic calm down signal. It directly dampens the activity of those aggressive effector T cells. TGF-β is also suppressive, but it plays a role in tissue repair too sometimes. That can lead to fibrosis. But its main job here is suppression.
And IL-35, that one sounds less familiar.
Yeah, it's a bit newer on the scene.
Yeah.
IL-35 is interesting because it doesn't just inhibit effector T cells. It can actually help convert other cells into regulatory cells too. So it kind of spreads the suppressive effect. Taken together, releasing these cytokines creates this local bath of stop attacking signals.
Okay, so that's the chemical warfare. Then there's the second contact-independent metabolic disruption. The sources use the term starvation, which sounds pretty direct.
It is, and it's quite clever. Tregs compete for resources. Remember we said they have high levels of the IL-2 receptor CD25?
Right, they soak up IL-2.
Exactly. Conventional effector T cells desperately need IL-2 to multiply and survive, especially during an attack. Tregs basically act like sponges, soaking up all the local IL-2, effectively cutting off the fuel supply to the aggressor cells.
Starving them of fuel.
And they do something else too. They can produce a molecule called adenosine. Adenosine acts like a brake on T cell metabolism and activation. So they're stealing the fuel and hitting the brakes.
Clever. But what if those methods don't work? What if some aggressor cells push through?
Then the Tregs can get physical, direct cell-to-cell contact. And they have two main ways of doing this. First, believe it or not, they can actually kill other immune cells directly.
Wait, the peacekeepers can kill?
If necessary, yes. They can use mechanisms similar to cytotoxic T lymphocytes or killer T cells, things like perforin and granzyme B, to eliminate particularly aggressive effector cells that aren't responding to the other suppression signals. Think of them as security guards who try talking first, then maybe restrict resources. But if things get really out of hand, they can take direct action.
Okay, that's surprising. What's the second contact method?
This one involves manipulating the other key players, especially the antigen-presenting cells, or APCs, like dendritic cells. These are the cells that normally show antigens to T cells to activate them.
Right, they present the wanted poster.
Exactly. Tregs use surface molecules, particularly CTLA-4 and LAG-3, to interact with these APCs. When CTLA-4 on the Treg binds to the APC, it basically tells the APC not to provide the necessary go signals, co-stimulation, that conventional T cells need to get fully activated. No go signal, no attack.
So they disarm the alarm system at the source.
Pretty much.
Huh.
And there's another layer. Tregs can also induce these APCs to produce an enzyme called IDO.
IDO, indoleamine 2-quasi-3-dioxygenase. That sounds familiar, linking back to starvation.
You got it. IDO breaks down an amino acid called tryptophan. Key cells need tryptophan to grow and divide. By making the APCs produce IDO, the Tregs essentially turn the APCs into tryptophan-depleting machines, creating a local environment where T cells just can't thrive. So across all these mechanisms, cytokines, metabolic disruption, direct killing, APC modulation, Tregs create this incredibly powerful multi-layered suppressive shield. #tag3, the balance in the allograft.
So these Tregs have this amazing toolkit, but for them to work in transplantation, they need to be where the action is in the transplanted organ itself, the allograft. Do they actually go there?
Oh, absolutely. They're very migratory. After a transplant, Tregs are drawn to the graft. They follow chemical trails called chemokines that are often released by the graft tissue itself. It's like a signal flare calling for regulation. So yes, we definitely see Tregs accumulating within the transplanted organ.
But the research seems clear that just having some Tregs there isn't enough, right? Because rejected grafts can still have Tregs in them. So what's the deciding factor between tolerance and rejection?
That's a crucial point. It's not just about presence, it's about dominance. The key factor seems to be the ratio of Tregs to the conventional effector T cells, the TEFs, the potential attackers.
So it's a numbers game. If the regulators outnumber the aggressors, things stay calm.
Essentially, yes, or at least exert enough influence. When researchers compare grafts that are successfully tolerated versus those undergoing rejection, the difference is stark. Tolerant grafts consistently show a much higher Treg to TEF ratio.
Okay.
And it's not just numbers. The Tregs found in those tolerant grafts often seem to be, well, better at their jobs. They show a more potent suppressive phenotype, meaning they're producing higher levels of those key inhibitory cytokines we talked about, like IL-10 and TGF-β.
So more Tregs, and the Tregs that are there are working harder.
Exactly. In contrast, in rejecting grafts, even if some Tregs are present, they're simply overwhelmed by the sheer number and activity of the effector T cells.
That ratio and the potency of the Tregs, that sounds like something you could actually measure and potentially target with therapy.
It is. And this isn't just theoretical, it correlates clinically. Studies show that transplant recipients who naturally have higher numbers of Tregs circulating in their blood or higher numbers infiltrating the graft tend to do better. They have better kidney graft function, longer graft survival, sometimes even respond better to standard anti-rejection treatments if they do hit a bump. It really confirms that Tregs are a key part of the body's own mechanism for accepting a graft. The whole idea behind Treg therapy is to artificially boost that ratio and potency to tip the balance decisively toward tolerance.
Okay, so the basic science is compelling. The animal models looked promising. Let's move into the clinic. How has this idea of adoptive Treg therapy, taking cells out, growing them up, putting them back in, actually performed in human trials?
Well, the initial results, especially from the early phase I trials, have been really quite encouraging. And perhaps most importantly, they've established a strong safety profile. These first studies, mainly in kidney and liver transplant patients over the last decade or so, showed that yes, this complex process is feasible in humans and it appears to be safe.
What does safe mean in this context, when you're infusing billions of live, specialized immune cells back into someone?
Good question. The key safety findings were no serious reactions during or immediately after the infusion, which is always a worry with cell therapies. Also, crucially, no dose-limiting toxicities, meaning they didn't find a dose level where the side effects became unacceptable. And maybe the most reassuring part, in the short to medium-term follow-up, there hasn't been any clear sign of an increased risk of infections or cancers linked directly to the Treg therapy itself.
That safety profile alone sounds like a huge win compared to the standard immunosuppressants, which we know do cause infections and do increase cancer risk.
It really does change the risk-benefit equation. And beyond just the absence of new problems, there's emerging evidence that Treg therapy might actually reduce toxicity by allowing patients to use less of the standard drugs.
Ah, so it enables immunosuppression minimization.
Exactly. One kidney transplant trial, for instance, reported that patients who received Tregs had 0 episodes of acute rejection over a 4-year period. And interestingly, they also had fewer opportunistic infections compared to a similar group of patients on standard drugs who had about a 21% rejection rate.
Wow. 0 rejection and fewer infections.
It strongly suggests that boosting the Treg population can allow for a significant, perhaps even permanent, reduction in exposure to those toxic conventional drugs. #TAG2. 4 key therapeutic approaches.
So it seems like the field generally agrees that boosting Tregs is the goal, but the how seems much more debated. The sources outline, 4 main strategies being tested right now, each with different levels of specificity and complexity?
That's right. There are distinct approaches under investigation. The first one is probably the broadest and logistically maybe the simplest to start with. Autologous polyclonal Treg infusions.
Okay, break that down. Autologous, polyclonal.
Autologous just means using the patient's own cells. We take their blood, isolate the Tregs, which are pretty rare, remember, and then expand them ex vivo, meaning outside the body in the lab. Polyclonal means we stimulate them to grow non-specifically, usually with antibodies like anti-CD3 and anti-CD28. The goal is just to get huge numbers, billions of cells.
So just flood the system with general peacekeepers, basically.
That's the idea. The big European 1 study really paved the way here. It showed it was safe and feasible to infuse these large numbers of polyclonal Tregs into kidney recipients. And importantly, they tracked the cells and found they maintained lineage stability.
Meaning they stayed as Tregs, they didn't switch sides.
Exactly. Their genetic programming, the FoxP3 expression, stayed stable. They didn't turn into dangerous effector T cells after being put back into the patient. That stability was key because it meant patients in the study could start to safely reduce their standard immunosuppressant drugs without triggering rejection.
Okay, that's the broad approach. What's next? Getting more targeted.
Right. The second approach aims for more precision. Donor alloantigen reactive Tregs, or darTregs.
Donor.alloantigen.reactive. So these are targeted specifically at the transplant.
Precisely. Instead of just expanding all the Tregs, here the cells are expanded ex vivo specifically in the presence of cells or antigens from the donor of the organ.
Why do that extra step?
The idea is to enrich the final cell product for Tregs that are already naturally programmed to recognize and suppress responses against that specific donor tissue. You're concentrating the firepower, so to speak, directly against the foreign transplant antigens, while hopefully leaving the rest of the immune system more or less alone.
Does it work better?
Well, Trials using darTregs, including some arms of that one study and the UK's TRAST trial, showed it's practical and safe. And the early data suggests that these targeted cells might be more potent, potentially allowing for a greater reduction or maybe even complete withdrawal of drugs like calcineurin inhibitors in some patients. It's promising.
Okay, approach number 3 sounds totally different. No ex vivo expansion at all. C, low-dose IL-2 therapy for in vivo expansion.
Yeah, this one tried to leverage the biology directly inside the patient. Remember how Tregs have high levels of the IL-2 receptor, CD25?
Right, they're IL-2 sponges.
The theory here is if you give very low doses of IL-2, the Tregs, being so sensitive to it, will selectively grab it and use it to multiply inside the body. So you'd boost the patient's own Treg population without needing all the complicated lab work.
That sounds almost too elegant to be true. Simple, targeted boosting. But I think you mentioned earlier this one ran into trouble.
It did. This is a really important cautionary tale in the field. While low-dose IL-2 does increase the number of Tregs circulating, at least one major trial in liver transplant patients had to be stopped because it unexpectedly triggered rejection.
Wait, triggered rejection? How?
It turns out that while Tregs love IL-2, they're not the only cells that respond to it, especially after a transplant. Aggressive effector T cells, if they get activated, can also increase their expression of the IL-2 receptor CD25.
Ah, so the low-dose IL-2 meant for the Tregs.
Ended up inadvertently fueling the fire, providing the survival signal to the very effector T cells they were trying to control. It caused this inflammatory cascade mediated by IF and gamma and actually made the graft more susceptible to attack.
Oof. That really highlights the need for absolute precision, doesn't it?
It absolutely does. It shows the danger of off-target effects, even with something seemingly targeted like low-dose IL-2. Which leads us neatly to the most precise approach currently being tested, borrowing heavily from cancer immunotherapy. CAR-engineered Tregs.
Like CAR T cells for cancer, but for tolerance.
Exactly the same principle, just flipped. Instead of engineering a T cell to kill cancer, you engineer a Treg to suppress rejection. You genetically modify the patient's Tregs to express a chimeric antigen receptor, or CAR. This CAR is designed to recognize a very specific target molecule found only on the donor organ cells, typically an HLA molecule.
So these CAR Tregs are like guided missiles, programmed to seek out the transplant and only turn on their suppressive functions when they get there.
That's a perfect analogy. The CAR acts like a super-specific antenna.
Right.
When the CAR Treg binds to its target on the donor cell within the graft, it gets a strong activation signal. This tells the Treg, okay, you're at the right place, deploy suppression now. Preclinical studies suggested these CAR Tregs, especially those targeting donor HLA molecules, could prevent rejection much more effectively, and with potentially fewer cells than the broad polyclonal approach.
And this isn't just theoretical anymore. These are in human trials.
Yes, this is happening right now. It's really the cutting edge moving into the clinic. There's the Steadfast trial, testing CAR Tregs targeting HLA-A2 in kidney transplant recipients. And the Liberate trial is doing something similar for liver transplant patients. The big question these trials are asking is, can this kind of ultra-precise donor-specific targeting finally allow us to protect the graft effectively while minimizing or eliminating the need for global immunosuppression? It's a very exciting space. #TAG3. Protocol refinements, the two-study example.
So it's not just about which type of Treg you use, but also how and when you give them. I saw the UK's TNOBO study Transplantation. Without over-immunosuppression, tried a different timing strategy. What was that about?
Right, the TWO study looked at integrating Treg therapy into the existing standard of care, but with a specific protocol. They used a T-cell depleting drug called lemtuzumab up front, right around the transplant. This drug wipes out a large portion of the patient's existing T cells.
Okay, so clear the decks first, then what?
Then, crucially, they didn't infuse the Tregs right away. They waited 6 months post-transplant to give the Treg infusion.
6 months? Why wait so long, especially after depleting the T cells?
The thinking was strategic. That initial depletion creates a window, a sort of immunological vacuum, called lymphopenia. The immune system then starts to rebuild itself. The hypothesis was that if you introduce the Tregs during this rebuilding phase, 6 months later when things are a bit calmer, you might preferentially skew the recovering immune system towards regulation and tolerance. You're giving the incoming Tregs a better chance to establish dominance as the system repopulates.
Interesting. So let the dust settle, then introduce the peacekeepers while the neighborhood is rebuilding. Did it work?
It showed real promise as a proof of concept. Patients in the TWO study who got this delayed Treg infusion were able to be successfully weaned off one of their two main immunosuppressants, mycophenolate, and were maintained just on low-dose tacrolimus.
So they cut their drug burden in half,
Okay.
essentially.
Pretty much. And importantly, they had stable graft function with no rejection episodes reported through 18 months of follow-up. It demonstrated that you can integrate Treg therapy effectively into existing protocols, maybe not to eliminate all drugs immediately, but certainly to significantly and safely reduce the overall immunosuppressive load. It's about finding that optimal integration.
Okay, the potential is clearly huge, but let's be realistic. The sources also highlight some significant roadblocks, biological hurdles, practical challenges. Let's dig into those. What are the big biological concerns?
Probably the number one biological hurdle right now is phenotypic stability. The issue is that Tregs, well, they aren't always stable. They can potentially change their identity.
Change identity. What triggers that?
The main culprit is inflammation. The environment right after a transplant is naturally inflammatory. Conventional T cells that might be trying to attack the graft release inflammatory signals. Cytokines like IL-6, TNF-alpha, IL-1. Under that kind of intense inflammatory pressure, some Tregs can actually lose expression of their key gene, FoxP3.
They lose the master switch. What happens then?
They can essentially convert. They stop being suppressive Tregs and can actually become pro-inflammatory effector cells themselves, sometimes called X-Tregs.
Wait, so the cell you infuse to prevent rejection could potentially contribute to it? That sounds like a major Achilles heel.
It is a serious concern. We've seen evidence of this instability, this conversion, in animal models, even in non-human primate studies, where some of the transferred Tregs lose their suppressive markers over time.
Yeah.
If the goal is long-term, maybe even lifelong drug-free tolerance, then ensuring these therapeutic Tregs remain stable, loyal peacekeepers, is absolutely critical. It's a big driver for the next generation engineering approaches we'll talk about.
Okay, stability is issue one. What's the second big biological challenge?
It comes back to specificity and off-target effects. We touched on this with the failed low-dose IL-2 trial. If you use the broad polyclonal Tregs, you run the risk of generally dampening the entire immune system.
Right, not just suppressing the attack on the graft, but maybe also suppressing important responses against infections or even developing cancers.
Exactly. That's why the field is pushing so hard towards donor-specific approaches like darTregs or CAR-Tregs. The ideal is to achieve suppression that's precisely targeted only at the graft, leaving the rest of the immune system fully functional to fight off pathogens and survey for tumors. The IL-2 experience was a stark reminder that even therapies that seem targeted can backfire if they inadvertently activate the wrong cells. Precision is paramount. #2. Clinical and logistics hurdles.
All right, beyond the fundamental biology of the cells themselves, there are huge practical hurdles to actually making this a widespread therapy. Moving from small academic trials to something patients can routinely access seems like a massive leap.
It really is. The logistical barriers are significant. First off, there's still no real consensus on the optimal protocols and dosing. How many billions of Tregs do you need to infuse? Is it 1 billion? 10 billion? Does it depend on the patient or the organ? And when is the best time to give them? Before the transplant, at the time of surgery, 6 months later, like the TWO study. We just don't have definitive answers yet. We need larger, well-controlled phase 2 and 3 trials to figure out the best practices.
And these Tregs aren't being given in isolation, right? They're usually added on top of existing immunosuppression. That must create really complex drug interactions.
Incredibly complex. It's a delicate dance. We know some of the standard drugs might actually work against the Tregs. Calcineurin inhibitors like tacrolimus are a cornerstone of transplant care, but they can potentially impair Treg function and survival because they block IL-2 production, which Tregs need.
So the main drug could be undermining the cell therapy meant to replace it.
It's a potential conflict. On the other hand, some drugs, like rapamycin, which targets mTOR, seem to be more Treg-friendly, maybe even promoting their survival and function. So, figuring out the right background drug regimen that supports the infused Tregs without causing toxicity or rejection. That's a major clinical puzzle right now.
And then there's the elephant in the room, actually making these cells. Manufacturing sounds incredibly complex and expensive.
It's probably the biggest bottleneck to widespread use. This is highly personalized cell therapy. It requires specialized facilities operating under strict good manufacturing practice GMP guidelines. The process starts with leukapheresis, basically filtering out white blood cells from the patient. Then you have to isolate the Tregs, which, remember, are a tiny fraction, maybe less than 10% of the CD4 T cells.
So you start with very few cells.
Very few. And then you have to expand them in the lab, nurture them, get them to divide over maybe 1 to 2 weeks until you have billions of them, enough for a therapeutic dose. All while ensuring they remain pure, functional, and stable. It's complex, it's labor-intensive, and yes, it's currently very costly.
That sounds like something only major academic centers could handle right now. It's not like prescribing a pill.
Not at all. And that complexity also introduces variability. Tregs grown from patient A might behave slightly differently than Tregs grown from patient B, even using the same protocol. Standardizing and streamlining this manufacturing process, maybe using automated selection methods, or developing better ways to feed and grow the cells is absolutely essential if this therapy is ever going to be widely accessible. #3. Safety and long-term vigilance.
Okay, despite all those challenges, let's circle back to safety for a moment. Because the data so far, at least in the short term, sounds pretty good.
It does. The short-term safety data has been consistently reassuring. Across the different trials and approaches, adoptive Treg therapy seems to be very well tolerated. We're not seeing acute infusion reactions, and critically, no clear signal of increased infections or cancers within the follow-up periods reported, which now extend out several years in some cases.
And you mentioned earlier, it might even improve safety overall if it allows patients to reduce their standard drug doses.
That's the hope, and there's some early data hinting at that, like the reduced infection rates in some cohorts. By minimizing exposure to drugs known to be toxic, Treg therapy could potentially lead to better long-term health outcomes, even beyond just graft survival.
But this is tolerance therapy. We're deliberately dialing down part of the immune response. That requires long-term vigilance, right? What are the theoretical worries down the road?
Absolutely. Long-term monitoring is crucial. The main theoretical concern is always the potential for oversuppression. Two key areas need careful watching over many years, even decades. First, could excessive Treg activity dampen the immune system's ability to conduct surveillance against developing cancers? It's a theoretical risk that needs monitoring.
Okay, cancer surveillance. What's the second?
The second is susceptibility to opportunistic infections, particularly chronic or unusual ones, maybe rare viruses. If the immune balance is tipped too far towards regulation for too long, could it create vulnerabilities?
So far, these long-term risks haven't really shown up in the clinical data.
Thankfully, no clear signals have emerged yet. But the follow-up time is still relatively limited for a lifelong therapy. So while the current safety profile is excellent, the need for ongoing, careful, long-term monitoring remains incredibly important. We need to ensure we're achieving that perfect balanced tolerance to the graft, but robust protection against everything else.
This is where it gets really exciting, moving beyond the current trials into what's next. The idea of engineering super Tregs sounds like science fiction, but it's aimed directly at fixing those problems of instability and specificity we just talked about.
That's precisely the goal. The next generation of Treg therapy is all about bioengineering, making these cells tougher, more reliable, and more precise. We know inflammation can make Tregs unstable, make them lose FoxP3, so researchers are using gene editing to basically fortify them against that.
How do you fortify a Treg genetically?
Well, one approach is simply reinforcing the command center, enforcing FoxP3 expression. You can genetically engineer the Tregs to overexpress FoxP3, essentially giving them an extra constantly active copy of that master switch gene. The idea is to make them inherently more resistant to losing their Treg identity, even when they're surrounded by inflammatory signals. It's like locking the switch in the on position for suppression.
Okay, boost the good gene. Can you also disable the bad pathways?
Yes, that's the other side of the coin. Researchers are identifying the specific signaling pathways inside the Treg that get activated by inflammation and contribute to instability. Then, using gene editing tools like CRISPR, they can knock out those pro-inflammatory pathways. For example, knocking out a gene called JNK1 in Tregs has been shown in experiments to make them more stable, survive better, and produce more of the suppressive cytokine IL-10. These edited Tregs are designed to be more resilient warriors for tolerance in that hostile post-transplant environment. #2, selective growth factors.
Let's revisit that IL-2 problem. The low-dose therapy failed because it wasn't specific enough. It boosted the wrong cells, too. How is bioengineering solving that problem? How do you make an IL-2 signal that only Tregs can hear?
This is a really elegant piece of synthetic biology. Instead of using the natural IL-2, scientists have developed engineered cytokines and receptors designed to work only with each other. The concept is often called an orthogonal IL-2 receptor pair.
Orthogonal, meaning they operate independently of the natural system.
Exactly. It's like creating a private communication channel. Here's how it works. First, you genetically modify the therapeutic Tregs to express a unique, custom-designed IL-2 receptor on their surface. This receptor is slightly different from the natural CD25. Second, you manufacture a corresponding altered IL-2 molecule, a synthetic cytokine.
And this altered IL-2 key only fits the custom lock on the engineered Tregs.
Precisely. The natural IL-2 in the body can't activate this custom receptor, and the altered IL-2 cytokine you administer can't activate the natural receptors on other T cells. So when you give the patient this engineered IL-2, it only stimulates the engineered Tregs to expand. You get selective boosting without any risk of activating those unwanted effector T cells.
That sounds like it completely solves the off-target issue. Has it worked in practice?
The preclinical results are very strong. This orthogonal system has been tested in mouse transplant models like heart transplants. It successfully expanded the engineered Tregs in vivo, and importantly, it led to long-term acceptance of the transplanted heart, even after they stopped all the conventional immunosuppressant drugs. It really demonstrates the potential for having safe, on-demand control over the Treg population in patients. #3 manufacturing and delivery enhancements.
Okay, so we can engineer better cells. But we still have that massive manufacturing bottleneck. Are there practical advances happening there too to make this process more robust or scalable?
Yes, definitely. There's a lot of work on optimizing the stability and purity during culture. For example, adding the drug rapamycin to the culture medium during the ex vivo expansion phase seems to help. Rapamycin tends to inhibit the growth of conventional T cells more than Tregs, so it acts like a filter, favoring the selective growth of stable, functional Tregs and potentially weeding out any contaminating effector cells.
So using drugs during manufacturing to get a better final product, what about the starting cells?
That matters too. There's evidence suggesting that starting the expansion process with naive Tregs, those are Tregs that haven't encountered their target antigen yet, identifiable by markers like CD45RA, might lead to a more stable and potent final product compared to starting with memory Tregs. Naive cells seem to have a greater capacity to expand and might be less prone to losing their suppressive function during the culture process.
And beyond making the cells better or easier to grow, what about delivering them more effectively? You mentioned flooding the system isn't always ideal.
Right. There's a lot of interest in localized delivery. Instead of infusing billions of cells intravenously and hoping enough get to the graft, why not put them directly where they're needed? Researchers are developing biomaterials like specialized hydrogels or tiny polymer scaffolds that could be loaded with Tregs.
Like a little Treg reservoir.
Exactly. The idea is you could potentially implant this device loaded with Tregs right next to, or even attached to, the transplanted organ during the surgery. This would create a very high concentration of suppressive cells right at the site of potential attack, hopefully maximizing the local effect and maybe allowing for a much lower total dose of cells needed, which helps with the manufacturing challenge too. #4. New sources of regulatory cells.
Given how rare Tregs are in the blood and how hard they are to expand, finding alternative sources seems critical for making this practical long-term. What other options are being explored?
This is a really active area. One promising source is the thymus. The thymus is the gland where T cells develop, and it's naturally packed with developing Tregs, often called thymus-derived Tregs or darTregs. In some surgeries, particularly pediatric heart surgeries, a piece of the thymus is routinely removed.
So you could potentially harvest Tregs from that tissue.
Exactly. Researchers have shown you can isolate large numbers of highly functional Tregs from this discarded thymic tissue. These darTregs might even be inherently more stable or potent because they're freshly minted, so to speak, and haven't been circulating in the periphery. In fact, the first in-human use of Tregs purified from thymus tissue was reported recently, which is a significant milestone.
Okay, thymus is one option. What about the ultimate goal, an off-the-shelf product?
That's where stem cells come in. The real game changer for scalability and accessibility would be generating Tregs from induced pluripotent stem cells, or iPSCs.
iPSCs, these are the cells that can be made from, say, a skin cell and then turned into any other cell type.
Correct. You could potentially create a master bank of iPSCs and then differentiate them in the lab using specific growth factors and signals into functional, stable Treg-like cells. If researchers can perfect this complex differentiation process and scale it up reliably,
you could theoretically have an unlimited standardized supply of therapeutic Tregs ready to go whenever a patient needs them.
that's the vision. An off-the-shelf Treg product would completely transform the logistics and economics of this therapy, making it potentially available to many more patients, much more quickly, and likely at a lower cost than personalized manufacturing for every single individual. It's still early days for iPSC-derived Tregs, but the potential is enormous. #outro.
So we began this deep dive looking at this really frustrating problem in transplantation, the need for lifelong toxic drugs and the long-sought dream of achieving true tolerance. We've journeyed through the science from those landmark discoveries by Sakaguchi, Brunkow, and Ramsdell that first revealed Tregs.
Right, through understanding their complex, redundant ways of suppressing immune attacks.
And then we analyzed the clinical strategies being tested right now, from just boosting numbers with polyclonal cells to the targeted darTregs, the tricky IL-2 approach, and now these really cutting-edge CAR-Tregs aiming for ultimate precision.
The pace of innovation, especially in the cellular engineering side, it's just remarkable. And the ultimate goal remains clear, achieving what's called operational tolerance. That means a healthy, functioning transplant, but with minimal, or ideally zero, chronic immunosuppression. Just the patient's own, perhaps therapy-boosted regulatory cells keeping the peace.
That would be utterly transformative for patients' lives, wouldn't it? Taking away that constant burden of drug toxicity and risk. It really feels like we're on the cusp of shifting from, broad chemical control to much more refined biological governance of the immune system.
It does. And maybe that leaves us with a final thought for you, the listener, to consider. We've seen how powerful Tregs can be at shifting the immune balance. But we've also seen they face challenges in stability, in inflammatory environments, the need for precision. The goal is operational tolerance, which kind of implies the immune system just learns to ignore the graft. But given everything we know about immune cell plasticity, the potential for Tregs, the theoretical long-term risks needing vigilance, does the immune system ever truly permanently ignore something foreign? Or is Treg therapy, even in its most advanced forms, fundamentally about establishing a highly sophisticated lifelong system of active monitoring and control managed by these engineered or boosted security guards?
That's a really interesting question. It's not about forgetting the graft exists, but about constantly, actively deciding not to attack it. How much monitoring is enough? How stable do those guards need to be to ensure tolerance and safety for decades?
Those are the questions driving the next wave of research. It's a fascinating challenge.
It certainly is. Thank you for joining us on this deep dive into the intricate world of regulatory T cells and the future of transplantation.